Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid, RNA, and proteins and not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S Swedberg's unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are the small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of the cilium or the flagellum showed that they are covered with the plasma membrane. Their core, called the exoneme, possesses a number of microtubules running parallel to the long axis. Microtubules and a pair of centrally located microtubules. Such an arrangement of exonemal microtubules is referred to as 9+2 era. The central tubules are connected by bridges and is also enclosed by the central sheath, which is connected to one of the tubules of each peripheral doublets by the radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structures called the basal body. Centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in the centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by the radial spokes made of protein. The centriole forms the basal body of the cilia or flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first discovered by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing, has highly expanded and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At the number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions and between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example, erythrocytes of many animals and sieve tube cells of vascular plants. वुड यू कंसिडर दीज सेल्स एज लिविंग? तो इसका एग्जाम्पल जो था ना, वन से ज़्यादा न्यूक्लियस किसके पास होते हैं, तो इसका एग्जाम्पल राइजोपस. द न्यूक्लियर मैट्रिक्स और द न्यूक्लियोप्लाज्म कंटेन्स न्यूक्लियोलस एंड क्रोमैटिन. द न्यूक्लियोलाई आर spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that interphase nucleus is a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells shows structures chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 23 pairs of chromosomes. You will study the detail of DNA packaging in the form of chromosome in class 12. Every chromosome visible only dividing cells essentially has a primary construction of the centromere on the sides of which disc-shaped structures called kinetochores are present. Centromeres holds two chromatids of chromosomes. Based on the position of centromere, the chromosomes can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the centrosome chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosomes, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Thank you.